Okay. Okay, so this is a biology lab for Mike Sands' class. I'm Emily Horton, and these are my two lovely subjects, Rosemary and Chris. Okay, so let's begin with talking about transcription, translation, and mutations. Okay, so the first thing you need to know is that DNA is composed of 4 nitrogenous bases, and I have them written here for you somewhere. Right. Where did they go? Right here. So there's adenine, and they will be represented by our purple clothespins. Guanine, they will be represented by our brown clothespins. For now, thymine, which will be represented by our green clothespins, and cytosine, which will be represented by our red-colored clothespins. Now, something to note is that adenine and guanine are both double-ring bases, while thymine and cytosine are both single-ring bases. And you need to know this because once we form our DNA strand, both our template and our complementary strand, you'll see in the middle what is called an alpha helix. And in between, in the alpha helix, you can only ever have 3 rings. Therefore, adenine and guanine will never bind together because then you would have 4 rings, and that would be too many. And thymine and cytosine can never bond together because you would only have 2 rings, and that would be too little. So keep that in mind. I'll give you guys that for now, so you can refer to that. So basically what I've drawn here already is this big circle on the Bristol board is your cell, and I've labeled it, and this in here is your nucleus, and this is where we will make our strands. Okay? So we can go ahead and start. What I'm going to have you first do is make your DNA template strand and your complementary strand. So we're going to use red pipe cleaner for this, and I'll just have you guys, you can just tie them together, and that will be, one of them will be your template strand and the other will be your DNA complementary strand. Yay, okay, perfect. Okay, so now before we begin anything else, I'm going to have you mark one of your strands with a yellow pipe cleaner, and that will represent your template strand. Because it's important to know, you have to know which is your template strand, especially when we start talking about transcription. So one of you can...
Just like at the end?
Yeah, you can just, I just wrap it around, like the whole thing, you can just wrap it around.
Like that?
Yeah. Perfect. That works. Or you can just
want the
even,
whole thing?
yeah, you can just like wrap it around at the end, yeah. You can just, yeah, what I do is I just like literally just wrap it around.
Okay.
So it's like all just at the end. Yeah.
Okay.
Yeah. It's just to identify which is your template strand because, like I said earlier, your template strand is that's where all of your DNA is. That's the information that we use. Whereas your complementary strand, we don't ever use any of that information on the complementary strand. So it's important to know that the DNA template strand is really the strand that's used, especially in transcription, and it's used in translation as well. Okay, so so now that we've done that, we are going to start with this sequence. So you can see it on here, and I'll give it to you guys for reference. But we're going to begin with making our DNA template strand. So I have some clothespins for you. And for now, ignore the black ones. So just put the black ones to the side. We will need them later, but ignore them for now. So if you guys want to go ahead, and using this sequence, I'm going to have you form your DNA template strand.
Do you want us to make the whole thing?
Yeah, so it goes A, G, T, and then all the way down to there. So you can just clip them on here.
Oh,
Yeah,
okay.
yeah.
That's how you want it.
As a strand, yeah. Yeah. So okay, so remember we're just making our template strand right now.
Not that
No, not that one.
one. Okay, yeah.
So all those clothespins will be
Yeah.
used just with this one.
Brown, right here.
Uh-huh. Right.
Yeah. Two more.
I guess they do kind of look blue, but they're supposed to be purple.
Okay.
Yeah. Two reds.
Thank you.
My hair's in here. My
That's
hair.
okay.
Purple.
And then two
Oh,
greens and a
okay.
purple. Yeah, sorry, it
There's
goes down there. I
purple.
couldn't fit it all.
Where's this one over?
Yeah, yeah. Perfect. So A, G, T, C, G, T, A, C, C, A, T, G, C, C, A, T, C, C, A. Perfect. Okay, so that is your DNA template strand. Now, keep in mind, it's just something to keep in mind as we go through this, but remember that adenine and guanine are both double ring bases. And cytosine and thymine, so the red and the green clothespins, are both single ring bases. So now our next step is we are going to form our DNA complementary strand. Now, you have to remember for this, and I have this here, but you're not going to look at it because I'm going to see if you can do it on your own first. Remember that A always binds to T. So your complementary strand is the opposite of your complementary strand is the opposite of your DNA template strand. So A always binds to T, so when you see an A there will be a T on the complementary strand, and C always binds to G. So your red clothespins will always bind to your brown clothespins, and your purple clothespins will always bind to your green clothespins. Okay, so knowing that, I'm going to have you form your DNA complementary strand. And I'll just grab these for now. Oh, whoa. Okay. There's not enough room.
So we're doing like the exact same thing.
Yeah, but remember, A binds to what?
T.
Correct? And C binds to?
G.
Perfect. So that's your template, so don't follow that one. I have the complementary that you guys can check once you're done. So if you line it up, yeah, and start at that end. So you have a purple clothespin and it binds to?
Browns to a T.
Perfect.
So
T.
purple.
Okay.
We need the purple one.
Nope, so you won't put the purple one, right? Because you already
okay.
have your template strand.
Okay.
So your complementary strand is
Is
just the opposite of
okay.
your template
So
strand.
then
So you have
that.
so what if you have a purple one? What are you gonna have on this
Green.
side?
Green.
Perfect.
Okay.
So yeah, you can just put it. I would just put it the other way so they kind of line up.
Okay.
Yeah.
And then so.
So you have a brown binds to.
C.
Right.
Right.
Red.
Perfect.
Right there. Got it.
Got it. Okay. Then green is A.
Correct. So purple. Yeah.
Okay.
Now purple is green.
Yeah. And then you have two reds.
Red is brown.
Correct.
So two browns.
There it is.
Now purple is green.
Yeah. Hey, you guys got it. So these two
Use the
strands that are
closed
forming,
Purple.
once these two strands have formed, in between is called the alpha helix, and that's where the rings would be.
Okay.
And you could only fit
Okay.
3 rings
Nice.
in the alpha helix,
Okay.
which is why adenine
fine.
can never bind to guanine and thymine can never bind to cytosine, because you'd either have too many rings or too little rings.
There it is.
So then that is two browns.
Sorry.
Now 3 grains. Purple. Yep.
Yep. Sorry.
2 browns.
I'm gonna move this for you.
Mhm.
1 green. 2 purples. And then green.
Perfect. Okay, so let's check it. So do you start with a T?
Yes.
Yes. And then you have a C?
Yes.
Yep. And you have an A,
Mhm.
and then a T,
Yep.
Yep.
and then 2 G's,
Yep.
and then a T, and then an A, and then a C, and then 2 G's,
Mhm.
and then 3 T's,
Mhm.
and then an A, and then 2 G's, and then a T, and then 2 A's. Perfect. Okay, so that's your DNA complementary strand. That's your DNA template. So now what we're going to do is we're just going to move them into, that all happened in the nucleus of the cell. So we'll just have those line up. Perfect. Okay, so I'm just going to talk a little bit about proteins because that's what we will be forming in translation, proteins. So proteins make up all of our cell structure. And they're made up of individual amino acids. So here we have, I've represented some amino acids. There are 20 amino acids in your body. 12 of them our body actually makes naturally. But 8 of them we actually have to take in through nutrition. So that's just a little bit about proteins. So now we're going to talk about mRNA and tRNA. mRNA is called messenger ribonucleic acid, so messenger RNA. And what that does is it carries, basically what it does is it photocopies a template, it photocopies your template strand of your DNA. And then the mRNA strand is where all of your amino acids are placed, and that's where translation will happen on your mRNA strand to form a protein or a protein chain. And then you have transfer RNA, which the tRNA has anticodons and your mRNA has codons. So your anticodons from your tRNA and your codons from your mRNA, they match up, they pair up, and then they form an amino acid. And then what the tRNA does is that's what drops the amino acid onto your mRNA strand to begin forming that protein chain. Okay? Okay, so what we're going to do right now is we're going to work a little bit with tRNAs and getting you comfortable with codons and anticodons. So I did a bunch of them for you guys just to save a little bit of time. However, I do have 4 here for you guys to do because I do want you to understand the process and understand how, if you have the codon, how to figure out what the anticodon is and how the amino acids form. So I do have a couple here for you guys to do. But for time's sake, I did most of them. Okay, so a key thing to remember is with tRNA and mRNA, in mRNA there is no thymine. So there will be, on your template strand, there will be no green clothespins. They will be replaced by our lovely black clothespins. And the black clothespin pins represent uracil, which is uracil will always bind to adenine. So black clothespin will always bind to your purple clothespin, and obviously uracil is a single ring base because adenine is a double ring base. And can you have more than 3 rings in the alpha helix?
Yeah.
Exactly. Okay, so looking at these, let's just go through one together. So this one I've done. And so you had your codon, which was UUA, and you're going to need this lovely sheet to refer to. You have your codon UUA. So let's locate UUA on here.
Oh, there's more over here.
Yeah.
I didn't see that. Right there.
Perfect. So it's going to be what amino acid? A little
black.
A little black circle, right. So you'll write in here little black circle, and then you'll take your amino acid from your bag and put it on top. And then what we have to do is we have to figure out what the anticodon is. So how to figure that out, it's really, really simple. What always binds with U?
Hey.
What is it? Yes,
Okay.
correct. So you're going to have A, and then what binds with A?
You.
U.
Okay.
So that is going to be your anticodon. It's what binds with what nitrogenous base.
Okay.
Do you sort of understand that?
Sort of.
Okay, so we'll do these, and you'll understand it more.
Okay.
So let's start. We can start with that one. So you have your codon, GUA.
Right there.
Perfect, and it's
Big black
Big
circle.
black circle.
so your amino acids. Now, I'll explain that after actually. Perfect. And
Anti.
you a pen to write with. Your anticodon would be what? So you have what always binds with G.
C.
Correct. Perfect. And then what always binds with U?
Okay.
Correct. And what always binds with A in this case?
U.
Correct. So that's your anticodon.
Okay.
So that's done. We can put that one there. So let's go on to the next one. So you have your codon AAG.
There.
Perfect.
Little red circle.
Correct. Perfect. So your anticodon would be what? What always binds to A?
U.
Correct.
So UU.
And what always binds to G? Perfect. So that's your anticodon. So that one's done. Put that one over there. And then, oh, that one rolled away. Perfect. We'll go into this one. You have your codon CCG. Or sorry, CGG. I read that incorrectly.
CGG.
Little
Beige.
beige circle.
Correct. And so what is your anticodon in this case?
So it'd be G.
GCC.
Yeah.
Correct. So write that in. Perfect. And then go on to the last one. So your codon is GUC.
Big black circle.
Perfect.
And then
Okay. So something to keep in mind, there is a stop codon. There's 3 stop codons, as you can see here. And the stop codon basically tells the protein chain to stop. So just stop making anything. And that forms a protein chain. However, we'll get to this later, but when there is no stop codon, what happens is all of your groups of 3 bases start to begin to shift, and that is actually a form of a mutation, but that's the very last thing that we'll talk about. So your stop codons it will never change. There will always be UAA, UAG, and UGA. And I actually have them written here. And you always have a start codon as well. And it tells the tRNA where to drop the first amino acid. So it's like, okay, start here. You don't necessarily start at the beginning of the chain. You start where you see you start where you see AUG. So I'll keep these here because I don't obviously expect you guys to remember that off the bat. And you'll notice that your start codon here is a big yellow circle, which is over there. And then your stop codon is in the bag. It's a big purple button. Yeah, so that is that. So we worked a little bit with tRNA and anticodons and codons. But now what we need to do is actually talk about transcription, translation, and then mutations. So we're going to start with transcription. And transcription is, what happens is the DNA unwinds. So you can imagine this is your DNA and it's all twisted.
Mm-hmm.
So what happens is the DNA unwinds. The complementary strand is pushed away. So we're going to take this away actually. We'll leave it, but we're going to put it to the side. And what happens is with the help of RNA, your DNA template strand creates a wonderful mRNA complementary strand. So we're gonna form that because I just want you guys to see how it works. And there's gonna be two parts. Remember what I said about mRNA? There is no thymine, so you will not find any green clothespins. So what do we have to do with our DNA complement or template strand? Change the thymines to uracils. So we can do that first, and then I'll have you guys create your mRNA complementary strand.
So do you want us to change it on here?
Yes,
Okay.
yes, I do. Yeah. So this is transcription. The DNA has unwound. The complementary strand has gone away, and now with the help of RNA, ribonucleic acid, it creates an mRNA complementary strand. And the mRNA strand is where translation happens and the protein chain forms. Perfect. Okay, so now with these two blue pipe cleaners, we will start to make our mRNA strand. And it's basically the same thing that we did with the complementary strand, except keep in mind, black will always bind to purple, and purple will always bind to black. That's the only thing that you have to remember. Everything else, C always binds to G, that thing. So I'll give you the clothespins, and you guys can work on that. So your first one is purple, and that will bind to
Black.
Black.
correct.
Okay.
Excellent.
And then it's brown, red.
Yeah. Perfect.
And black. I'm thinking that right? Yeah.
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 So green.
Sorry, I realize
Got
that
it.
purple and black are pretty similar.
Got it. Purple.
Black.
Black.
Correct.
And
Red is brown.
brown and one more. Purple, so
Black.
Yeah.
Is that black?
Yep.
Okay.
Yep. Now that's black.
Then black is
Purple.
purple.
Come on.
Brown is red.
Correct.
Red. Red
Brown.
is two brown. Yeah. And then another brown.
Purple is black.
So you have 3 purples.
And then purple.
Purple. Brown. Two.
And then purple is black.
You guys got the hang of it.
Green. No, I mean purple.
Purple.
Yeah,
Purple.
you got it.
And then
Perfect. So, you guys just completed transcription.
awesome.
Yay!
Aced it.
Okay. Okay, so what we're going to move on to next is what's called translation. And translation is the formation of single amino acids, and the amino acids are dropped along your mRNA strand to eventually form, once you reach the stop codon, it forms a protein chain. And you have a protein.
Yay.
So that is why I have all of these tRNAs with the amino acids on them. Because what we're going to do right now is, first I want you to locate your start codon, so AUG, and we're going to be working with the mRNA strand. So we can just move this back up a little bit. Just picture that it's still in the nucleus. So find, you're going to start, that's going to be your starting end.
This one?
Yes. So find AUG. So it's going to be a purple, black, brown.
Right here.
Perfect. So, using your sheet, what is your AUG, your start codon?
It's that big yellow.
This yellow.
So it's, yeah, so all of them that we're going to be using are up here, so you can, if you want to put that down. You can put that, voila, the tRNA brings it over and drops it. You can just put it on top of the codon. Perfect. So you have your first amino acid, your start codon. So now we're going to look. Codons are always groups of 3 bases. So one clothespin is a base. So a codon will always be 3. So that's how we're going to read the next ones down the strand. So you have your start codon, and then you have a brown, a black, and a purple. So it's going to be a what is it? It's a GUA. So actually, you can look for them
the
on
big
here.
black circle.
Yep, you can look for them on here as well. The tRNA brings it over, drops the amino acid. Perfect. Okay, so let's look at the next one. So you have a red, brown, brown. So if you want to find them on here, so you have a CGG. So you're looking for your codon because codons are on mRNA and anticodons are with tRNA. Perfect.
Oh.
So your tRNA brings it over, drops the amino acid. Voila. Okay, so that was that one. So now you have is that L?
Three.
No.
Oops.
Yeah.
Yeah.
Wrong way.
Wrong way. Okay. So your next one, you have 3 blacks, so you have U, U, U.
Okay.
So we're looking for U, U, U. Or you can also look for it on there to find it faster. Perfect. Yep. So your tRNA brings it over, whee, and then drops it off. Perfect. Okay, so what's your next one?
AGG.
AGG.
Right here.
Yeah, perfect. Beautiful. So now what you have, what's your next one? UAA. Now, keep in mind, what did I say about
It's the end. It's the
It's-
end. Stop.
perfect?
Yeah.
So, actually, that is still in the bag here. It is your stop codon.
There's a big purple button.
Yes. I had to make it a little different. So I'll let you put that on there.
Oh, that's awesome.
And you have now made a protein chain.
Yay.
Because there is a stop codon there, so it has told the tRNAs and the mRNAs to stop matching up because the protein chain has been formed. Okay, so that is transcription. It's basically the formation of single amino acids, and they are brought, the codons from the mRNA match up with the anticodons on the tRNA. They come together, they form an amino acid, and then the tRNA drops the amino acid on your mRNA strand, and eventually it forms a protein chain. That is translation. Yes? Do you guys kind of get it?
Yeah.
Yeah? Okay. Okay, so what we're going to move on to now are lovely mutations. And mutations occur for mostly two reasons. The first reason is mistakes were made in the making of your DNA. So what happens is your body checks and rechecks and checks again as your DNA is copying for any mutations. But however, like life, like things in life, mistakes do happen, right?
Yeah.
And that's the first reason why or how a mutation can occur. The second reason, and the more common reason, is environmental factors. So things such as known carcinogens, so UV rays, radiation, chemicals such as smoking, those also cause mutations as well. So what we're going to be looking at today are 2 separate mutations, and they are single base mutations. So remember, like I said earlier, a single base is 1 clothespin. So we're only going to be looking at single base mutations. And the first mutation that we're looking at is something called point mutation. And for me, the easiest way to think of a point mutation is it's a substitution. So you're taking out one base and you're substituting it with another. That's the easiest way to think about a point mutation for me. So we're going to actually demonstrate it on the mRNA strand and the DNA template strand. So what we're actually going to have to do with this strand is we're going to change the blacks back to greens. And where did my bag go? Here it is. And actually, you know what? We're going to use the complementary strand as well. I'll do this one. lovely complementary strand. Okay. We're mostly going to be working with the mRNA strand still, but just for visualization, we'll use all 3 strands. Okay, so now we're going to locate on all 3 strands our 7th base. So from the beginning, we'll start 1, 2, 3, 4, 5, 6, 7. So purple. So your purple.
1, 2, 3, 4, 5, 6, 7.
So
Just
black, right. So your black will be replaced with the purple. So you'll take that out, yep, and you can just put it to the side. Your purple will be replaced with a green, and my purple will be replaced with a green. So your 7th base has been substituted. One single base has been taken out to be replaced with another. Now, for right now, we are only looking at our mRNA strand. So what was that? Was on there, right?
Yep.
There you go.
Okay. So now let's locate our start codon, which is AUG. So I'll let you guys find that.
Which would be here.
Correct? Right. So now that doesn't change, does it?
No.
No. So let's look at our next one. Our next one was, so it should have been, it was a brown, a black, and then a purple. But what is it now? It's G.
G-A-A.
Correct. So let's locate GAA. Where is it? There.
Yeah.
Found it? Perfect. So what happens in point mutation is the amino acid completely changes in that codon.
Okay.
But now let's look at everything else. Does anything else on the protein chain change?
Yeah.
Exactly. So in a point mutation, the single base is substituted, and that codon, that amino acid, completely changes. But as you can see, nothing else on the protein changes. Like no other amino acid changes. That is a point mutation.
Okay.
Okay? You guys
Yes.
kind of get that?
Yeah.
Okay. So let's change our bases back to what they were. And we'll change the amino acid back.
So that
Yep.
Black. And then we'll put, it was the big black circle, right?
Yep.
Okay. And you'll change that one back. Should be a purple, right? 7. Okay, so the last thing we're going to talk about. You can all do a happy dance. The last thing we're going to talk about is a frame shift mutation. And it sounds weird, but it's pretty simple actually. For me, the easiest way to think about a frame shift mutation is it's the addition or the subtraction of a single base pair. So if we look, so 1, 2, in this case we're going to use our 8 base pair. So 1, 2, 3, 4, 5, 6, 7, 8. So the green is just taken out. Nothing is substituted, it's taken out. Or in another case of a frame shift mutation, another single base pair can be added. But for this exercise, we're going to use subtraction. So locate your 8th base pair on your DNA template and your mRNA complementary, and just take it out and leave the space. Yep. Perfect. Perfect. So now we're going to look at our mRNA complementary strand. So let's locate the start codon again. Perfect. And what is it?
AUG.
Perfect. Exactly. Okay, so we have AUG. Now let's look at what you had before. Ooh, I need that. What you had before was a purple, right?
Yep.
Yeah.
So it should have been GUA. In a frame shift mutation, that single base pair is removed if it's a subtraction. So now let's look at, what is it? It's G. We now have to shift down, right? So it's GUC.
C.
So no longer, that amino acid changes. So where's GUC?
Is it that one? No.
The codon.
Yeah. Wait. G. Oh no, that's G. Same thing. Why is it on
GUC?
there? No, I'm looking at the same thing.
Oh. That's because it's right here.
Oh, okay.
And what is it? GUC is what?
Oh, there it is. Where is it? Oh, big black circle.
Right. So it becomes it becomes a big black circle. Instead of G,
Yeah,
it would have been
instead
GUA
of-
instead of that, right. Okay, so now let's look at the next three. So you have GUC. Now what you have, again, everything is shifting, right?
Yeah.
It's a frame shift mutation. So now you have GGU. So where is, I see it.
The white circle.
Yeah. Perfect. So you're going to pick up that amino acid, and now that changes. Just put that there. Okay, so now do you see how everything is shifting down the
Yeah.
line? Because everything downstream of where the frame shift mutation happened always changes. But you notice that the start codon, because it's upstream from the mutation, it's not affected.
Yeah.
So now as we're seeing, everything is changing. So we'll keep going because you'll need to see why. So that was, what was that?
That was the
G.
GGU. Okay. So now we have UUA. Where is UUA?
Right here.
The codon?
UU.
That's the
it's a
antibody.
little
Sorry.
black circle.
Correct. So that amino acid is brought over, that's
So
dropped.
you're reading upside down.
I know. And that changes. And then what's the next one? We have GGU.
Little white circle.
Right. So we can grab another one.
Oh.
Yeah. So that beige changes to a little white circle. Perfect. And then what do we have next? We have AAU.
Big
So
orange circle.
perfect.
And it keeps going, right? 'Cause it's not the end.
Exactly. You no longer have a stop codon. So in a frame shift mutation, you notice that where the mutation happened, everything downstream from it, everything shifted one, and that changed every amino acid completely. And then there is no stop codon, and so the frame shift mutation just keeps going. So some real life examples of frame shift mutations would be HIV, cancer, because obviously the cancer cells just keep growing. There's no stop codon to tell them to stop growing.
Okay.
So the cells just keep on
That makes sense.
replicating, yes. So what did I say? I said HIV, cancer, and then cystic fibrosis is actually another common example of frame shift mutation. Okay? And that is all I have to talk about today. That is protein synthesis. So we talked about transcription, which is the formation of the mRNA strand by the unwinding of the DNA. Then the complementary strand is pushed away, and with the help of the RNA polymerase, the mRNA complementary strand is formed. Then we talked about translation, which is on your mRNA strand. It's the formation of single amino acids through the reading of the mRNA codons with the anticodons on the tRNA. They come together to form an amino acid, and then the tRNA drops the amino acid off on your mRNA strand and to form a protein or a protein chain. And then the last thing we talked about were mutations, why mutations occur, and the two single base mutations were the point mutation and the frame shift mutation. And that is all I have to talk about. Do you guys have any questions about what we talked about? Did it help?
It
I
did.
think so.
actually,
Did it?
yeah.
Visualizing it
Yep.
and actually like moving the amino acids and that kind of thing?
For me, this is like the best way to learn. I have a learning disability, so it literally
This kind
I
of thing
saw
really
it
helps.
and it helps. I understood everything.
Perfect. Awesome. Okay, so I think that is everything. And I will be signing off now.